we can start. Very good afternoon to all of you. We have with us Professor Rohan Williams. He's a computational biologist based at Singapore Center for Environmental Life Sciences Engineering, SCELSE, a research center of excellence co-hosted by National University of Singapore and Nanyang Technological University, where he is the head of the Integrative Analysis Unit. Professor Williams also holds an appointment as a principal investigator at the Singapore MIT Alliance for Research and Technology, SMART. His research interest lies in metagenomics and multi-omics analysis of microbiomes and complex microbial communities. Previously, he was a research group leader at the Australian National University from 2007 to 2011 and an NHMRC Peter III Fellow at UNSW from 2003 to 2007. He holds a PhD in human clinical physiology and a BAPSc in physics. So with this short introduction of Rohan, I would now request Rohan to start with his talk. Thank you so much once again for accepting our invitation and sparing your valuable time. Thank you.
Great. Thank you very much, Professor Mahanti, and good afternoon, everyone. It's a real pleasure to give this presentation here today, and I hope the meeting is going successfully. So today I'm going to talk about what is called genome-resolved metagenomics. So this is basically the kind of the sort of contemporary way that metagenome data or DNA sequencing data that is obtained on microbiomes and complex microbial communities is sort of analyzed. And in particular, I'm going to focus on the new long-read sequencing technologies that have emerged in the last 5 years. I just need to disclose that I'm a co-founder and equity holder at a Singaporean biotech company that is involved in this area. And sorry, I just need to move my slide. And this is my research group at SCELSE. It's called the Integrative Analysis Unit, and I call it a unit because we're not only a research lab, we also do a sort of bioinformatics service provision and bioinformatics training, and so we have quite a mix of activity. But our primary focus is on analyzing the composition and function of complex microbial biofilm communities. And we use a mixture of mostly dry lab or data-driven approaches, but increasingly in recent years, we've used wet lab approaches as well. And this is the group in somewhat happier times before we have to wear a mask all the time. These are some of the techniques we use, and today I'm going to focus on metagenomics and the analysis of that data. So I imagine most of you will know what a microbiome is, but for those that don't, it's really just a way of capturing the microbial species, so bacteria, archaea, viruses, fungi, some fungi, and microeukaryotes tend to live in these kind of fairly organized, fairly structured communities, and so they're interdependent on one another. You can see this example here where these sort of filamentous bacteria and these sort of circular bacteria kind of tend to coexist together. And so this is not a new realization. I mean, we've understood this for a very long time, but the study of microbiomes has really been given a big push with the development of technology over the last 15 to 20 years. And I should also point out that, of course, microbiomes can be host-associated, living within or with plants and animals. They are, of course, an incredibly important part of the natural world, so aquatic and marine environments, soils, sediments, but also in air. There is a distinct microbiome in the air that we breathe, and there's been a lot of work done on this over recent years, and also in engineered environments. And today I'm going to mostly talk about wastewater treatment systems, but also things like cheese rinds and other biotechnological or engineered environments that are very heavily dependent on the activity of microbiomes. So, so wastewater treatment is really an a sort of essential essential infrastructure that developed economies need to use. And so this is an example of a wastewater treatment plant in Singapore. The input to this is really sewage from mostly from human waste. And it gets processed in these tanks, and I mean, I imagine the environmental engineers in the audience will know this this area very, very well. If you blow up these tanks here, they're these treatment systems, which are really a kind of clever combination of bioprocess engineering and biological activity. And if we look inside these tanks, there is a complex microbial community seen here. This is a fluorescence in situ hybridization image of the biomass in such a community. And if we blow it up again, we can see down to individual species. And so in SCELSE, there is, over the last 10 years, my colleagues and I have tried to build a kind of interdisciplinary research program that is oriented towards understanding the microbial ecosystems biology of these wastewater treatment communities. And we make use of data from field studies by sampling full-scale operational plants. We make use of bioreactor studies where the microbiome in these communities is used as an inoculum in these sort of chemostat-style bioreactors. And more recently, some of us have got interested in culture isolates of different kinds. My own group has become particularly interested in Diplococcus vannus as a model system for some of the bioprocess ecophysiology that occurs in these plants. And this is a very interdisciplinary activity. I mean, we have, these are the kinds of expertise that are represented in this work. Environmental engineering, Stefan Wirtz and Yingyu Law and Eze Santillan, sequencing and some bioinformatics, Stefan Schuster and Daniela, and then metabolomics, Yulan Wang, imaging, Peter Tarak, and then environmental microbiology and other areas. And I'll talk more about some of the collaborative ventures later. And then again, these are some of the techniques that we heavily rely on. So it's a very interdisciplinary, very mixed, kind of environment, which is exactly the intention. This is the reason that SCELSE existed, to enhance this sort of activity. So if we go back to the wastewater treatment plant microbiome, I'm just going to use this I mean, this is a very artificial example, but it's for those of you that are perhaps not familiar with this area. We can. I mean, this is a ridiculously simplified example. Just assume there are two types of bacteria in the plant. The problem with most of these bacteria is that we don't know how to culture them. So this is true for most natural environments. We really have no idea how to culture most of the bacteria that we find, and therefore we can't use the sort of repertoire of genetic and biochemical and physiological, morphological techniques that classical microbiology developed very successfully. So we need to use other approaches, and these are so-called culture-free methods. And these culture-free methods are basically reliant on analyzing the DNA sequence of the entire microbiome. So the idea is that you extract all the DNA that you can from one of these samples, from one of these plants, and analyze that DNA and try and understand the composition and function of the community. And this, of course, is very dependent on technology. So here is a timeline of developments in the history of DNA and genome sequencing. The sort of the sort of 25 years or so from 1965 to 1990, where a lot of key technologies were developed, including the the development of Sanger sequencing, which has been described by some commentators as a sort of silent revolution in biology. At the time, it wasn't quite clear exactly how impactful this would be. And then this next period where the human genome was sequenced, and then after that, kind of a bunch of other kind of important genomes were sequenced. The first microbiome studies were done around here in around 2004, 2005. But what really happened was that was important was the first commercial high throughput DNA sequences, like typified by the Illumina instruments. And then after that, there was kind of an explosion of activity, including more recently in sort of massive kind of microbiome surveys of all kinds of microbiomes on Earth and this kind of thing. And then more recently, a set of technologies has emerged which really give us a lot of power to reconstruct the genomes of member species of microbiomes. So how is this work oriented? Well, we're interested in genome-resolved analysis. So we extract all the DNA from one of these samples, we sequence it on one of these instruments, and in the case of Illumina, we end up with many, many, many short DNA reads. There's many ways this data can be analyzed, but I think the field is converging on the use of assembly-based methods as a kind of like a standard. So each of these short reads is combined by a computer algorithm back into slightly longer stretches of DNA, reconstructed stretches of DNA that are called contigs. And then if you're lucky, you can start piecing these slightly longer fragments together into longer ones again that we call scaffolds. And the idea is that you take scaffolds which under a set of analytic criteria are likely to arise from the same genome and reconstruct an entire genome. And that's, in an ideal world, this is what it will look like. It would be a continuous chromosomal sequence. It would have all the right kinds of ribosomal genes, or the requisite number of tRNA genes and so on, and you'd be able to sequence all the repeat regions and construct a single sequence. But in reality, it's not that simple. You have contamination from other genetic material from other genomic contexts. You have missing bits of genomes that you can't really reconstruct properly. And you also have multiple contigs, so often very, very fractionated genomes. But even so, this data is still useful, of course, because once you've got that genome sequence, you can predict genes, you can obtain their protein sequence. These days, especially, it's with AlphaFold, it's increasingly straightforward to make predictions of protein structure. And then you can start putting all this together into these so-called kind of systems biology models of metabolic and regulatory pathways. But it's still a very difficult process in the context of a metagenome analysis or microbiome analysis. So in this plot is trying to give you some idea about how complex that is. For example, this is a single sample from a bioreactor, enrichment bioreactor. There's about 53 million short DNA reads that are generated, which can get assembled into about just under 400,000 contig sequences, and they're plotted on this diagram here. Each data point is a contig sequence or a scaffold sequence, and here we're showing the coverage, or just how abundant that contig is, and its GC content, just how many G and C nucleotides it has. And this is a very useful measure to try and separate pieces of DNA fragment that are coming from the same genome. And so we can form these kind of, the details of this I don't have time to go into, but we can form these working models of genomes, right? So they're very fractionated, they have multiple contigs, but under a given analytic model and hypothesis, we can say that they're likely to mostly be from the same genome. And the way of doing this now is very standardized. It's almost a push button procedure, but of course you still have to look at the results. And it's particularly difficult when you're dealing with really complex communities. And this is a point I'd like to make about wastewater communities, is that they are very, very complicated. So this diagram here comes from a beautiful paper in Nature Microbiology a couple of years ago. That's a consortium that analyzes wastewater treatment samples from all over the world with 16S sequencing. And we can see here that this is on a logarithmic scale, roughly the number of, some surrogate measure of the number of species in the community. And we can see that the wastewater treatment plants, WWTPs, are more complex than the human gut or animal gut microbiomes by at least kind of one order of magnitude, often more. So they are very, very complex communities. And actually they don't have much in common with human feces. It's a completely different set of bacteria that are in play here. And they're feeding off the waste that humans generate, but they're a completely different set of bacteria to what's in our gut. And they tend to be more similar to the kind of species you see in freshwater or oceans or soils. So which is perhaps not surprising, but still definitely worth documenting. And so using this kind of genome-resolved analysis, we can study these communities. This is a paper we've recently studied, which is a collaboration as part of this collaborative paper as part of this overall research program, led by Mindia Haryano and Kritika Aramagan on the analysis side, and my former colleague Yingyu Law sort of led the collection of the data and the wastewater engineering aspects of this. And we can see this is a phylogenetic tree of the high quality genomes that we can obtain from these samples. There are 24 samples in total in this analysis, but we can see that here in this figure here, we're basically breaking down the genomes we're capturing by how abundant they are. This is the genomes that are in the top 10% of the abundance range, the next 20% and so on. And we're just counting the proportion of high quality or medium quality or low quality genomes. And there's a standard set of criteria that are used to categorize how good quality these genomes are. And we can see that the high quality ones tail off very, very quickly as you move down, as you get less and less abundant in the community. For a given sequencing depth, it's very, very difficult to capture these genomes. And that just reflects the overall complexity of the community. It's a very, very complex system to try and analyze. In total, we generate about 9,000 genomes, and these are redundant because we're analyzing multiple samples. There's the same genome is often recovered multiple times. There's about 3,000 non-redundant genomes in total. Only about 1,000 of these are roughly high quality, and far less than that approximate something close to a complete closed genomes with all the right genes and all the right ribosomal genes that should be there. And we can do another interesting calculation if we look at the entire metagenome assembly and just pick off 16S sequences, count the number of them, and that's if you remove the redundant sequences, that's some approximation to the number of recoverable species. And we just when we compare that to the number of genomes we capture, we estimate we can capture about 60% of the community with this genome-resolved analysis. And we estimate that's consistent with roughly there are about 5,500 species in this community. So it's a very, very complex sample. That's over an order of magnitude higher than the complexity of the human gut microbiome. And we can do other kinds of analyses with this. We can of course pick off individual species. That phylogenetic analysis lets us do that. And here, for example, we're comparing different genomes of a species called a species in a genus called Candidatus accumulobacter. And this is a key species for the preferential accumulation of phosphate from wastewater streams, which is an important chemical to remove, but also an important chemical to potentially recover, because phosphate mining is becoming increasingly difficult. and here we can see different species of genomes of different species of Accumulobacter from both within Singapore and also in neighboring countries. And so some of these species we can enrich in enrichment bioreactors, but some of them we don't see enriched in bioreactor experiments. And so there's an interesting question about why that is. But with the genome sequence now, we may be able to start to approach that problem, at least from one angle. So if I can just summarize what I've said so far, using short read data, this approach of obtaining metagenome assembled genomes, or MAGs, it lets you recover kind of a working model of a genome. Illumina sequencing has a very low error rate, so that's good. But there's really a lot of limitations, and the big ones are that the recovered genomes are still quite fractionated, and so that has a lot of implications for the analysis of genome quality. And the overall approach is also very dependent on how complex the community is, both in terms of the sheer number of species, but also what their abundance distribution looks like, and also how much genetic variation there is at a subspecies level. So if there's a lot of strains of the same species, you're not gonna get very good results from this kind of approach. And also identifying horizontal gene transfer is very, very challenging because of the fractionated nature of the genomes. You don't know whether a short contig is a contaminant, or is it a genuine piece of genetic material from another bacteria that's been transferred into the bacteria that you're recovering that genome from. So what's kind of changing this game a bit is long read sequencing. And so Pacific Biosciences, Oxford Nanopore Technologies, these are single molecule sequencing instruments. If you extract a DNA from a cell or cellular preparation and you put it into one of these instruments, you will actually sequence the individual bit of DNA. You won't have to go through these kind of complicated procedures where the DNA is broken up into little bits and then PCR'd and so on. You're directly sequencing what's there. So it's a very, very exciting development. And surprisingly easy to use. I mean, these instruments are big. Oxford Nanopore, they're very, very compact instruments that can, literally fit it. For those of you who've not seen these, they're very, very small. There's also a kind of interesting set of so-called synthetic long-read technologies, which use a combination of short-read sequencing and molecular barcoding techniques to basically make longer sort of synthetic reads. And 10x Genomics is one company that does this, and there's a new kit, or it's a new sort of, I'm not sure, it's something between a kit and a platform by Illumina called Illumina Infinity, which I think will also be an exciting kind of synthetic long read approach. And then there's also these techniques that are based on chromosome conformation capture, which were originally designed to understand the chromosome structure of eukaryotic nuclei, but they've basically been adapted to metagenomics to try and capture pieces of DNA from within the same cell. Phase Genomics is a startup company, actually pretty well established now, that has kits for using these techniques. So there's a lot of exciting new technologies coming out in this game. And what we're really aiming to do is get up here. So this is read length. This is like 10,000 base pairs per read of DNA. And we really want to get up into this region where we're generating a lot of these kind of reads. And if we could get there, then at least we start to have a chance of reconstructing large amounts of genomic material from these communities. The problem with this is that until very recently, these long read technologies had very, very high error rates. So back in 2015, I think when Oxford Nanopore was, if you'd asked me at the time, I would have said this is very much a sort of toy technology and it wasn't serious. And then I think the flipping point was kind of somewhere in here. All of a sudden, the error rate or the accuracy started increasing. And it's been an up and down, situation. But now in 2022, the error rates are really very, very low. So this gives it a tremendous advantage. And we started working with Nanopore back, I think, in 2016. We did our first runs towards the end of sort of September, October 2016, and I was very, very skeptical. I mean, I very skeptical that this would work. And part of the reason for that was that because of this high error rate. And so this is a paper we did with Daniel Hussen from Tübingen, who's a longstanding colleague and collaborator of mine. And, we could get these long reads from a MinION sample from metagenome samples, simple ones from enrichment reactors with probably about 100 species in them. And we could actually assemble them into contigs. And in fact, the first sequencing run we did actually generated a complete genome of the most abundant member in the community, which kind of stunned me. I was like, wow, this is super exciting. But the problem was we really couldn't do any more with it because the error rate was so high that we couldn't predict any genes, that the gene prediction algorithms didn't work properly. We couldn't use any of the genome quality assessment tools for the same reason. So Daniel came up with a very clever idea, which was to use data from NCBI to basically try and correct the frame shift errors. And this let us apply these tools and make assessments of the gene content and then the genome quality. And that was very exciting because from as little as 1.2 gigabase pairs of MinION data, which took about a day to generate on one of these benchtop instruments, we were able to capture complete closed genomes from the top 7 members of the community, which was really, really exciting. And those genomes just literally dropped out of the assembly algorithm in one piece, which again, I was blown away by. I thought this is really, really exciting stuff. We've subsequently sort of extended this work. This is work led by Irina Bessarab and again, Kritika Ramagan, who's one of my sort of key bioinformatics people in the group. And we've been able to capture complete closed genomes from these major functional species that contribute key functions to wastewater treatment, but also important structural bacteria like Theothrix. And I'm going to talk about this a bit later, but we've also been able to capture non-chromosomal replicons, which are very, very difficult to reconstruct using short read data. And so we can convincingly capture plasmids and phages, but also things like chromids, which are kind of multi-part genomes. I mean, some bacteria have multiple chromosomes, and we can actually seemingly we can capture these. And also some of these new small bacterial genomes from the candidate radiation phyla, these recently recognized novel phyla groups that have been basically captured by metagenome analysis. And so if we move ahead a bit, this is another project that I'm particularly excited about. This is the ASEAN Industrial Wastewater Genomics Consortium, which is co-founded by myself and Adeline Chua from the University of Malaya and 2 colleagues from Bangkok, Benjamin Suraraksa and Perida Pramanate, who are from KMUTT in Bangkok and also are at a government research institute there. And the idea with this consortium is that we're going to use these genome-resolved techniques to study, obtain genomes from microbiomes that are involved in agri-industrial waste treatment in ASEAN countries. And this is a big area because of like the palm oil production and other industries. And I think these industries are also very important in India as well. So what we've done so far is worked on systems like this. This is a USAB bioreactor. It's an anaerobic digester treating wastewater from a citric acid production plant. We've been able to capture complete high quality genomes from both bacterial and archaeal methanogenic species in these systems, and then start to sort of maybe tease apart the functional basis for competition between these species that types of species that impacts biogas production. And then some of that work's been done with long-read sequencing as well. So the results I described previously were from simple enough microbiomes that we could capture like the key species in complete closed form from with long-read sequencing. But when you move to a more complex community like this, it's not that simple, right? So again, this is this plot that breaks down for different kinds of metagenome assemblers or assembly workflows and data types. It breaks down the number of high-quality genomes we're getting, which is not very much. 15 high-quality genomes if we use a mix of short-read and long-read data. The best result from long-read data alone is 14 genomes. But if we look at the short-read data, we capture like 70 high-quality genomes. They're very fractionated, they're very broken up into bits, but we can still capture more of them. And what this suggests is that it's not going to be as simple as just generating long-read data and all these genomes will drop out. We will have to, again, construct so-called genome binning algorithms, which are a very prominent feature of short-read genome-resolved metagenomics, to try and recover genomes from long-read metagenome data. And this is one of our efforts. This is one of our efforts here in that direction, which is sort of getting ready for submission. So what's the take-home? What's the summary of all that? I think long-read sequencing in metagenomics will be a key current and future technology. I think in the next 6 months, the next year, you will see a massive amount of data getting generated using these techniques. But I think it's always important to bear in mind that genome data, by itself, for these sort of problems, doesn't mean anything. I mean, it needs to be embedded or means its utility is limited. It needs to be embedded and contextualized with other process engineering and other biochemical and analytical chemistry data to really be useful. And the implications of that are very big if it's done properly. Genome data can also identify previously unknown microbes. I think there are massive advances that have been made in this problem in the last 5 years due to some of the new bioinformatics techniques that have been developed. And secondly, and this is the consortium will also focus on this, is what secondary products can be obtained from this genome data. Obviously things like new FISH probes and other molecular probes, but these new long-read sequencing technologies are so easy to use that in principle they could be deployed in the field very easily. And I'm involved in some other projects, and people in my group are involved in such projects in medical and aquaculture analyses, which are making great progress in this, but I haven't got time to talk about them today. What I am going to finish off with is talking about how long-read sequencing technologies, and I have a few minutes left, can be used to study this important problem of antimicrobial resistance, which, as we all know, I think is emerging as a massive kind of public health problem on a global scale. And it's a very complicated problem because of the complex interrelationship between human and natural ecosystems, and also our agricultural and animal husbandry procedures basically drive this problem. So what we've done in this is a collaboration that was Krasi Hystrova from Marquette University is the lead PI on this, but we've provided an analysis of these plasmidome extractions from E. coli isolates. So Krasi and a group kind of basically isolate E. coli from different human and natural ecosystems and perform DNA extractions which bias towards the plasmidome being extracted. And we can sequence these on MinION. I probably don't have time to talk about this plot, but it's basically trying to use, so we can assemble this data and reconstruct a relatively small number of contigs. And here we're just trying to combine the results from different programs to try and understand what they are. Separate out the chromosomal fragments that are reconstructed from viruses and plasmids and the kind of plasmids that they are. And we can actually capture complete closed plasmids from these samples, and we know that we can compare them to databases of very, very well studied plasmids, and we can show that we can pick off things like this F-type plasmid G150, which comes from one of the most important strains of E. coli that's expanding across the world, strongly implicated in the propagation of antimicrobial resistance. And we're also using these techniques with colleagues from NUS and NTU this is an island off the coast of Singapore that's used by recreational humans for recreation and for work, and it's also close to aquaculture sites and shipping lanes. So it's a really interesting kind of potential model system for thinking about microbial resistance and how it propagates between human and natural ecosystems. So to summarize all that very quickly, I apologize for the speed I ran through that. We can use long-read sequencing to capture non-chromosomal replicons from these E. coli isolates from natural environments. We can understand what sort of plasmids they are. These conjugative ones are very well studied. There's a small proportion that are mobilizable. The majority are non-mobilizable as defined by current understanding. And it's actually sort of really difficult to figure out what these things are. And I'm not an expert on plasmids or chromosomes or plasmids or phages or anything like that. I'm just not that kind of genomic biologist. And it surprised me how complicated this was. It's actually very, very difficult to tell whether you're dealing with a plasmid or a phage or a virus in some ways. And then, of course, I saw papers like this